Should we just get started, or are we still waiting for people to connect? Yeah, let's just get started. Yeah. So just, I think, folks who were not there last week, I think the next 2 weeks, classes will be on Zoom, and then lab sessions will be exactly where we have lab sessions. I'm in the field right now, so it's midnight for me, which today is a really tough one because I jet lag and everything. I haven't had the chance to sleep yet, but I think today I'll sleep well tonight. But I appreciate.
Where are you today, Manu?
I'm actually off the coast of Naples in Italy, and we have just this week started kind of a sampling session off the coast here to catch a bloom that's happening right now. And many of the instruments that you saw, we have 3 squids and 2 gravity machines here at the Marine Station. And one of the goals is to be able to measure the dynamics of carbon flux in the ocean as it happens. And the time that was planned almost 2 years ago, but because of COVID got stalled. But now we are very close with lots of people coming together to try to make these measurements. So I think, yeah, maybe next lecture, if I'll share some pictures and threads, at least in the lab session, to show you guys what we're actually doing. The coast is right here, and it turns out this town, there are random fireworks in the middle of the night, which just happened 10 minutes ago. So if we get interrupted a little bit, you'll hear a whole bunch of fireworks. Okay, so I'm gonna get started. Tyler, can you give me permissions to share my screen? We're gonna continue on the thread on art and science of paper, but today we wanna cover much more on the diagnostics front and things that we didn't discuss. Before we get started, I'm curious if somebody cracked the chirality puzzle that we discussed last time, or if you have a good handle on thinking about the chirality experiments that many of you did. And then the other part would be is do try to share your calculations that you're all trying to do for the specific projects that you're choosing. And then, of course, the same thing goes for creating. Some of you might not have been in the lab session. We discussed the Fred Park framework. I have to dig in. I've been trying to find some old notes for you guys so that I could give you many other examples of that framework used for other projects. I use that. I just learned it from friends. I first heard from that framework from a mechanical engineering professor at MIT that I took lots of classes, Alex Slocum. It's buried in his textbook on precision machine design, and I couldn't find any other good reference for it. So I'll try finding an electronic version for that, or at least my old notes of some examples. If you search for it, you get my graduate assignment that I had submitted 15 years ago. On Google still remembers it, and that's the only reference to it, which I'm a little confused by, but I know I'll find it. But I think the idea is that everybody for their projects should try to make sure that you create a Fred Park sheet and try to iterate that a couple of times. Any questions on that? Any kind of confusions on that front? Otherwise, I'm going to get started. And again, I think this is supposed to be interactive. If you can't hear me, let me know. Just don't see many of you, so I just want to make sure that folks can. So should I just assume that folks have had the chance to test that framework and people are comfortable with it?
And sorry, Manu, when you say the framework, are you referring to the Fred Park?
The Fred Park framework. Did anybody apply it to their projects as yet? Any questions around that? Anybody got stuck?
I think so far there's a chance. So I didn't list it as one of the homework things for today. So there's a chance folks haven't done it yet. I'll make sure to put it on the
let's put that as a homework for this week. And again, all of you know that I mentioned these things at homework. This is not something that anybody is grading. Your primary grade will be your projects, your final projects, and your team projects are what are actually graded. But again, if you want to develop this as a technique, it's actually very valuable to practice this for a whole bunch of things. You'll realize it opens up far many more windows than you might think you might be closing based on the risk analysis. So please try to do that. And then again, do it for the projects that you're actually passionate about. Okay, so I think, let's get started. Let's talk about much more of the chemical and the diagnostic sides of things today. We covered last time quite a lot in terms of the mechanics of paper. And I want to flip a little bit around the chemistry of paper, but really focus on diagnostics. The lab this week will be where we'll be opening up a whole bunch of diagnostic devices. So it's valuable if you can collect as many COVID tests as possible, especially if you could collect some of these new class of COVID tests that are PCR equivalent. These are called LAMP assays. I'll have a few that I'll pass to Anesta and a couple people in my lab. We'll share a little bit about a COVID test that we've been developing, but one of the key aspects of the lab will be a breakdown of many of these diagnostic devices. So you can bring any kind of diagnostic device. You could bring pulse oximeters, you could bring other diagnostic devices that you feel comfortable breaking and are low cost, all the way from low cost thermometers to any of these lateral flow assay tests. And then we will have a few for folks to play with, but it's very relevant to the discussions that we will have now. So I think let's start with may or may not have heard of. And again, going back to the context of frugality, of course, I'm assuming people have used nitrocellulose and chromatography paper. So most paper fluidics, which we will talk quite a lot about, is essentially made out of this specialized paper called nitrocellulose. It's standardized in some sense. And I think one of the factors that you have to think about if you will be starting working with diagnostics front is that the paper.
Okay. Hey, Manya, I think we lost you for a second there. Is there a chance you can turn off your video maybe?
Yeah, maybe I'll try turning my video off. Can you still hear me? Can you hear me now? Yeah. Okay, we'll try without a video for a second. But one of the things that I was just mentioning was, as you are thinking about designing a diagnostic device, one of the key important bits, and especially if you're using the class of materials, the materials have to be incredibly standardized. One of the key materials that's used in that space is these nitrocellulose membranes, or what are called chromatography papers. They're fairly cheap, but there is another class of a paper that I just want to bring up. These are called FTA papers, and you can find them commonly. This is currently manufactured and made by GE, and one of the fantastic thing in terms of the class of this FTA paper is really thinking a little bit about a standardization around both lysis and protein denaturation and simultaneously trapping nucleic acids. But again, on frugality, if you were to go look for these FDA elute microcards, which are very common in assays when you want to collect a genomic sample out in the field, for a pack of 25 cards, that's roughly around $100. And one thing that you should think about is alternatives to these types of papers that have actually been proposed in the literature. It would actually be valuable if any of you are thinking about a diagnostics project in this space to find. Can you all still hear me?
Yep, sounds good to me.
Okay, yeah, somehow I lost the window. So let me try. I don't understand this, that I do not see any of you. Something suddenly changed. Okay, now it returned back. Okay, yeah, this is much better because I wasn't sure that any of you guys are still there. One of the things that historically is fascinating, which happened quite a lot in the context of COVID, have had the chance to break open and think a little bit about the steps that are involved in, say, a PCR test that's done for COVID. And if you remember early on, one of the key sets of reagents when there was a first spike of COVID was these RNA extraction kits and DNA extraction kits were missing. So many places had all the primers, but they could not run the tests because they did not have the sets of materials to essentially first extract clean samples from, say, either the nasal swabs that were collected. And, going back historically, sometimes, of course, the classes of techniques that are currently used in large-scale assays are these magnetic beads. But this is a paper that I pulled out, which is just an absolutely stunning paper from 1944. And this has led to a paper-based approach for DNA and RNA extraction. It's the paper from Princeton by Alberts and Ferris and others. And one of the things that they discovered accidentally at that time is that cellulose is a remarkable material for capturing, because of the bonds between DNA and cellulose, for capturing an enrichment of DNA and RNA. And it was an accidental discovery at that time, but that led to a framework of thinking about how you could do enrichment assays just purely, literally using paper. And since that discovery in the 1940s, of course, this was done in a context of a basic science, and it still is really a mystery for why the molecular chains, for example, in cellulose bind to DNA so well. There is a very clear data around this, but the mechanism is still kind of if you look at the bottom in that picture, you can see the cellulose and you can see DNA, and then there is this nitrile bond with the cellulose. And this is the effective mechanism that people thought at that time. Now, the irony is that this mechanism had been known since the 1940s, but DNA extraction remained an expensive technique because of just the way it was implemented in the past, until I think in 2017, 2016, this really remarkable paper came out, which demonstrated that you can actually do a nucleic acid purification from a very broad range of organisms. They demonstrate of course animal samples, microbes in less than 30 seconds. So I'm going to walk through you this assay because this is an assay that any of you can actually do. And one way of thinking about this assay is, if you look at these Eppendorf tubes on the right, I'm going to zoom in. You start with a sample. So in this tube, you're seeing primarily a lysate from a plant leaf, for example. So you can see this chlorophyll, you can see a liquid. You literally a paper strip in this for 3 seconds, and you dip that out, and then you dip it for 3 seconds in a wash buffer. And what that's doing is that, of course, now the DNA that was lysed, you have to mechanically lyse your sample. So you want to shake it in some way and form that the cells would break. The moment you put in that lysate, any floating DNA or RNA would bind essentially to cellulose. And literally by picking that up and putting that in the wash buffer, what you're doing is you are removing extraneous proteins and other things that might have also come along. And literally 3 seconds later, you put that just a single dip in an amplification reaction buffer. So this is where you would say, for example, if you want to do PCR or you want to do LAMP, you are now transferring some of that DNA that was bound, which is pure because it's very specific binding to paper, and then you close the tube, and then essentially you run an amplification assay, and as good
Oh, sorry, Manu, we lost you for a second there. Would you mind repeating that?
And this assay, you know, is here.
I think we lost you a bit, Manu. Ooh, big time. All right, we'll give it 1 sec for him to jump back on. But in the meantime, I guess any questions or things that got jumped over when he was talking, we can help explain in the meantime while he's figuring out his Wi-Fi. These can be anything on any of the chromatography stuff that we've just been looking at. All right, looks like Manu's back on.
Yeah, Tyler, can you hear me now?
Yes, I can.
Okay, yeah, I don't know what happened. Just Zoom just crashed. Maybe I should close a whole bunch of programs.
Yeah, maybe your bandwidth does sound a little bit low still. So
because
yeah, Manu, I think your connection is still a little
try
bit.
Moving from one location to another location.
Okay, sounds great.
Okay, I'm gonna try just moving. Give me 2 seconds. Yeah, I think I might be a little bit distant from the router. Tell me if that changes the connection. I just have to figure out. So is this a little bit better?
Yeah, so far it sounds quite good.
Okay, I think that might have just been the case where all I need to do now is to figure out that I can still have power. Okay, let me know if this is better.
Yes, sounding great so far.
Okay, all right. I'm gonna reshare. Sorry about that, everyone. I haven't tested out this connection yet. So any questions on this, the extraction assay for a second? I think one thing that I would really recommend is I'll share these slides with all of you. Take a look at this paper. If you're really interested in the context of diagnostics, it's really a fantastic paper for a couple of reasons. One of the factors is that it's utilizing a scientific fact that was known literally for the last 60 to 80 years, and nobody had utilized it in a practical context, but the scientific basis for this was known. And secondly, for whenever you do field work, it becomes very valuable if rather than collecting the samples, you can only bring the extracted nucleic acids. So for example, currently, many of the malaria trials that happen carry blood spots on pieces of paper because much of these bound nucleic acids maintain And even when they're completely dried at room temperature, you can bring in to an extent that not only can you detect, say, a sequence of malaria using PCR, many folks are actually able to also sequence malaria using these dry blood spots. So it's really a remarkable technique that's used in the field very broadly, associated with environmental sampling itself. And then the other part of this is just the ease of use, because primarily the number of components involved are very limited. There are a couple of downsides of this as a technique, but one thing to think a little bit about would be is utilizing a chemical property of a given process and in terms of being able to run these sets of reactions. So this was much more about extraction. I'm going to take
a quick question on this. So as compared to the COVID diagnostic reagents that everyone's running out of, is this like, are you looking at this as an interesting approach that would be potentially viable in that situation, or is that just completely different because you have a different proteins in the fluid?
Yeah, no, not only is this potentially viable, one of the tests that I will talk about little bit later, we actually use this technique for extraction of RNA in COVID saliva samples. So remember, this is a two-step process. When you collect a complex sample, say saliva or nasal swab, you have many proteins in those samples that might actually interfere with your amplification assay. So many of the enzymes that are amplifying the sequence you're interested in might be inhibited by salivary proteins. And so it becomes important that you do the extraction and purify. And actually a test that I will show you in the very end, at least in the context of the work that we did in COVID, but this was also shown by a couple other groups as well. You can directly use this technique to build a COVID assay, while the big shortage that happened on the extraction assays that are used regularly in PCR reactions are based around these magnetic bead-based assays. And again, some of the costs that we pay for in our amplification assays are around extraction reagents. So I think this is very directly relevant to anything that's trying to do a type of a test that nucleic acid based. Of course, there are better chemistries. You can even bind DNA to many other surfaces, even stronger than paper. But one of the key things to think a little bit about, which we will talk when we quantify how diagnostic tests are quantified with the WHO criteria. So I'll pick that up again, which is how do you choose between access and sensitivity? And where would a technique like this fit, while where would a technique like PCR actually be valuable? Yeah, you should all interrupt me. Otherwise, it'll kind of become a little bit like a monologue. So I want to talk a little bit about malaria for a second, because I already mentioned the genotypes screening for malaria. I think I had mentioned in the past before about this plot at the bottom. This is a plot of number of cases per year, reported cases of malaria around the world from 2010 to 2017. And one of the unexplained trend in this context is the rise of cases since 2015. I don't have the data here for 2018, 19, 20, and all the way till 22, but this trend has slowly continued. And the thing that I just want you to take away from is literally when we say around 200,000 cases were detected, these are all diagnostic tests done per year. This is what's the reported data. So of course, there are lots of unreported cases of malaria. But the key worrying trend associated with this is the rise of cases. There are no really good explanations. I think some countries are doing really well in terms of elimination campaigns, while other sets of countries are faltering dramatically. So these rise of cases are occurring in pockets rather than occurring all around the world simultaneously. But having said that, there are many case studies and papers around increase of encatchment areas for malaria because of climate change. And so, I don't think there is a direct thread as yet, but needless to say, the most important point here is that the low-hanging fruits in malaria, which is the bed nets and some of the common things that have been done for the last 50 years, we have been able to reduce this curve of malaria going down. And for the first time, at the least, it's flattening. There are no low-hanging fruits anymore, which is a challenging situation. And that's when you would think about, where does diagnostics play a role? And I think this is a really fun plot in terms of thinking about how diagnostics has already played a big role. And you can also think about this in the context of economics itself. So 2010, WHO came up with a very firm guideline that nobody should be treated with malaria pills without a diagnostic test. So that's a fascinating stand that they took, because primarily malaria, there is resistance. So many of you know, we discussed artemisinin resistance last time. And one of the challenge here to think about is that is our frontline drug, which now there is common resistance genes in malaria. And both predicting this, there was a very ambitious program in trying to think about ramping up RDTs. I don't have the most recent data, but I think this number has definitely crossed around 350,000 tests per year. But this is the ramp up all the way from 2011 all to 2014, where there is roughly around 300 million tests done per year. Just so to keep this in mind, we roughly have around 500,000 deaths per year. And I had mentioned last time, in terms of target of what can diagnostics do, we are talking about roughly 2 billion people that have to be diagnosed every year for you to saturate diagnostics. So many of you who have thought about COVID, you can think about that if you don't diagnose enough number of people, you'll never be able to actually tell what the current infectivity rates look like. So at this moment, between 500,000 tests are done every year. I would say maybe somewhere around 40% of them are microscopy, but majority of them are these RDTs. And the irony is that roughly the cost of production, which we will talk quite a lot about, can range anywhere from 20 to 50 cents, while most RDTs, at least the latest ones, are accessible at a list price of somewhere between $4 to $5 to even $10. And of course, there are certain sets of programs that subsidize RDTs. And so several countries across Africa, many of these RDTs are subsidized. And so the question to think a little bit about is, what could be done? Is there a way or a framework to first think about, can RDT costs be lowered? And where are our challenges when you were to ask in terms of, do we need new ways of thinking about the diagnostics in this framework? And so I think one thing that I just want to make sure that everybody knows and is clear, if you want, you can read the original lateral flow immunochromatography assay, which is also called LFAs, the original patent. And the original patent utilizes something very, very simple, which is paper is a fairly interesting substrate in which you can pattern your antibodies, you can pattern any sets of your reagents that will act as your readout mechanisms. And you can use capillary wicking rather than any pumps or valves for the fluids to transport. So if you look at that patent on the left, I would say in the field of diagnostics, this patent is probably the most interesting patent. And I don't know if this is the original patent, because the last patent I could find is only 2002. So if you look here, my intuition was that there should have been more patents in this space, and there must be. So if somebody can find the original lateral flow assay patent, that'll actually be really fun. But just to give you a context, and this is what you will all open up. This is an example of a pregnancy test. And you can see that you have a sample. you have certain sets of antibodies. There is a test line, and then there is the substrate that you're really trying to detect that leads to this sandwich assay that forms. And the sandwich assay is what allows for you to be able to detect whether something has those antibodies or not. And you can see that the antigen is in there. And one of the contexts to think a little bit about is that on the left side are these gold beads that the liquid is picking up when it's passing through. And then there is a Vic pad on this side, which you will see when you open up these devices, which acts as a way for a lot of liquid to be able to pass through. So when we open these devices, you'll get an intuition for thinking a little bit about how much volume should pass through a given RDT or a lateral flow assay test. And I think you will get a very good handle of this when you open it. We will do this for Abbott tests, a couple of pregnancy tests, but it's valuable to think a little bit about how far can you push this framework, especially in the context of cost. So I want to pull out to show you just how these lateral flow tests are actually made. And then maybe another kind of an important anecdote to remember. I'm assuming at this point, all of you have done these tests yourself.
I'm working.
So I'm going to show you just a little bit about how these tests are manufactured. Let me pull this out to skip the ad. So this is really the completely automated platform for manufacturing for these tests. And what you're going to see is essentially a robotic pipeline. I'll just let this play for 2 seconds.
Manufacturing. They provide manufacturers flexible production capacity within a desktop footprint. Vision-guided component placement, strip cutting, and quality control ensure a consistent...
So literally, if you have seen desktop 3D printers, you should think of 3 desktop 3D printers capable of producing a lateral flow test in a completely modular fashion anywhere. So one of the interesting things about this is such a mature technology at this point that you don't need that much capacity or infrastructure.
Bulk feeders are not product specific and fit a wide variety of cassette types. The lateral flow test strip is fed with single card magazine, or reel and feed modules.
So one of the things you can see there is that there is a carrier, there is the actual strip, there is reagent loading, and then essentially it all gets packaged into the final carrier.
Is completed before assembly.
But what I love about this is that, when you think about manufacturing for diagnostics, you might be thinking of these giant factories, and literally a fairly small scale setup can allow you to manufacture these in a completely automated fashion.
Checks to make sure the lateral flow device meets all manufacturing requirements.
And, since COVID happened, most of you have essentially played or looked through these sets of devices. One as a challenge that I just want to mention, which a team last time picked up, which is most of these LFAs actually use plastics for their carriers. And that plastic is not that functional per se. And there was a project last year, as I said, roughly 500 million of these are done just for malaria alone. So if you combine malaria, pregnancy, and several sexually transmitted diseases, 10 to 20 of these, you're thinking about 4 to 5 billion of these tests that are done. And of course, they're single shot disposable plastics. And one thing to think a little bit about would be is, could you actually use substrates that are biodegradable in the context of creating the kinds of cartridges? So Tyler, looks like my visuals got lost again. Can you guys all still hear me?
Yep, you sound good to me.
Okay, yeah, I don't know why the visuals keep going in and out. Okay, so I'm gonna kind of broaden out this diagnostic discussion. Does anybody have a question so far on the two properties of papers that we just discussed? One is the binding and the other is the absorption. I wanna kind of zoom out and give you guys a flavor of what WHO recommended a couple of years ago for anybody who's interested in designing a new diagnostics framework. There is a beautiful kind of a hierarchical structure to follow. So I'm going to dive in a little bit in that structure. So unless anybody has a specific question on RDT manufacturing, and then we'll cover a lot of these RDTs when we are in the lab, because then all of you will get a chance to play with them. So any questions so far? Otherwise, I'm going to talk a little bit about a WHO policy framework for what is a good diagnostic test. So we'll kind of zoom out a little bit out of paper. I want to do a little policy discussion, and then we will come back and talk a little bit about, again, the technology side of the story. So anybody, I'm curious, has anybody opened a RDT test before? Have you been curious before you throw it, that COVID test that you've all done now, probably hundreds of, to actually open to see what's inside it? I'm curious if it matches what I just showed you. I mean, of course, if you open an RDT, it's not valid anymore. So don't read your test out. But Diana, were you going to say something?
I did open it. It looks the same, just a strip of paper. Yes.
Yeah, and I mean, one of the things a little bit about this, I made that comment about manufacturing. I found it shocking when COVID numbers picked up, the number of malaria RDT supply in Africa started going down. So I was a little bit confused. So we started digging of what is the connection between COVID RDTs and malaria RDTs. And it actually turns out Abbott, which is the company that made the BNX Now test, essentially captures 90% of the African market for malaria RDTs. And none of them are actually manufactured in Africa. And so when the demand on them and the pressure on them on producing COVID tests increased so much, they essentially took their malaria plants and they repurposed them to make COVID RDTs. And that caused a massive scare in the malaria community because Abbott has such a large footprint. And again, this is the direction where much of global health diagnostics and vaccines are moving towards, is the fact that because we don't have on the ground capacity, you can end up having these funny effects where, why should it be that a company that is far away, where there is no malaria cases, owns 90% or more of that market? Or the fact that there are no reliable manufacturers that are currently supported in Africa for malaria RDTs. And, I think, of course, there are many complicated answers to this, very much like vaccine inequality that we'll talk about next week. One important factor to remember is it's not technology, because as I showed you, literally a desktop printer can do something like that. And it's a complex framework of not investing for a long period of time in capacity building for manufacturing. Not anticipating the fact that if the markets are in a certain place, then we should really also be manufacturing locally. And so this is one of the key challenges in diagnostics, is you can design globally, but you really have to be thinking about increasing manufacturing capacity locally for these things to be both cost effective and for it to not be disrupted in the context of supply chains. Was there a question? Okay, so I wanted to mention this because I find it kind of appalling in many ways that majority of malaria tests are really made completely outside of the framework of countries that suffer with malaria. One really big challenge that's happening, you might think that applying diagnostics is a very, of course, since WHO mandated diagnostics for malaria, it has been remarkable because we have been able to treat people for what they are sick. But there is a downside that happened, which is when you have 500 million people being detected for malaria using similar RDTs, you can cause resistance that comes because of the diagnostic test. And one of the biggest scary situation right now in malaria diagnostics is that there are 2 gene deletions that have been detected. One is the PFH-RP2 and a PFH-RP3. These are what are used in the diagnostic test. And so the parasite realized that they were not essential. And because they were not essential, the parasite was able to evade them. And so now, because we are primarily using this as a diagnostic test, most people that have that gene deletion were not detected. And then this mutant essentially spread. So to a point, all across Africa at this point, both of these 2 are incredibly common. And it's quite dangerous because these patients show up as malaria negative on a test while they have all the clinical symptoms that are associated with malaria. So as a clinician, you are put in a very difficult spot because you would like to be able to follow WHO guideline in terms of thinking about what to actually do. Like, you should always only treat people when you have a diagnosis, while on the other hand, it is challenging because now with these gene deletions, unless you have a proper diagnostic test that can capture these as well, you are essentially putting the patient at jeopardy, and you're also actually expanding the scope of these sets of gene deletions. And this is also why the earlier discussions that we had about microscopy, that microscopy cannot be written off because microscopy is agnostic to many of these types of mutations that will happen in the genome of the parasite itself. And then many people currently are working on new class of malaria RDTs that depend on essential genes. One of them, for example, is around the context of thinking about any of the essential functions that all the parasites need to do. If you design an RDT trying to detect that, then it is very unlikely that the parasite could evade it because it would be an essential life cycle function. And so you can think about many sets of things around cell division, just common functions that no parasite can survive without would be a better target. And you can go back and ask this question, why didn't we do this before? And this is things that we can only do in hindsight, but it's very ironic that we found ourselves in this situation because of large-scale deployment of diagnostic tests. So sometimes too much of a good thing can actually be a bad thing. Okay, so I think we're going to dive into this very large-scale sort of field that has been developing in the context of what is now called paper-based diagnostics. And I'll kind of present this. Much of this is discussions with another friend of mine who had presented last year, Kevin Land, and he had a very nice review paper covering much of the policy discussions that I'm going to talk about. If any of you are interested, I'll send you this paper itself. It's fairly easy to find, but it's a very good review of the entire field. But the biggest thing that I just want to mention is if any of you are picking up a diagnostic project, this is a WHO framework. It's called Assured. It came out essentially a couple of years ago. And if you are building and designing a diagnostic test, you really need to be thinking about affordability, how sensitive and specific it is, which is, of course, everybody talks about. Avoiding false negatives is sensitive, avoiding false positives is specific. User friendliness. This is one that is often, as a designer, you might think about at a later stage, but it's incredibly important, even to an extent that many of the times the tests that are done in RDTs for COVID, the mistakes are actually made by the users themselves. Of course, there is this context of rapid and robustness. E stands for equipment free. And then the deliverables are really around thinking about how do you get a test to the people that need them, which might sound so obvious, but there are lots of COVID tests that are out there that are in hands of people that either don't need them or have the funds or support and resources, while there is, on the other hand, very large scale populations that actually need tests and have no tests. So, I think each one of these criteria has to be put in place if you're thinking about kind of a context like this. I think we discussed malaria. You can apply a similar kind of a framework to HIV in terms of the numbers, for example, for what you really need to do. There is a very large class of unmet needs, of course, in the context of contaminants. So when you're thinking about diagnostics, it's important to not just think about human samples. You could also just be thinking about samples that are in the water. And I know I put this slide in because I wanted to make sure that I know Santiago, Valerie, Ayusha, and a couple other people are thinking about water contamination. It would actually be valuable to see if some of you can find RDT tests that are associated around water contaminations and outbreaks. I mean, of course, cholera and a couple other ones are really good, but it would be valuable to think about whether there is an analogy that you can apply from the context of either human samples, but then apply it to detect for environmental samples. And then I think I discussed this last time. I was talking a little bit about vectors. The same sets of diagnostic technologies that you could be thinking about for humans could also be applied for the vectors that transmit these diseases. So just like you can detect a pathogen in a human causing a disease, you can also detect that same pathogen in a host that is transmitting the disease or is the vector. And then again, as we discussed about mosquitoes, you would need to reduce the cost even further for the diagnosis in the ecological setting to be viable. And you would have to reduce that cost by 1,000 to hundreds of thousands or even 1,000,000-fold, just because it is so much harder to really find the environmental context associated with many of these sets of diseases itself. Kind of another framework which is not captured so well might be captured in this last criteria that we discussed, which is the deliverables, or who are you designing for, is really around firstly understanding who is the client for your test that you're building. So most LMIC countries, low and middle income countries, have a very classic healthcare system. So you can think about, I had called for when I was thinking about Liberia and the Ebola discussions that we had, which was building tests for the community, which is called the 0th level. Community health center is where there is just an ASHA worker or a community health worker, and there is no facility. The layer above that is the primary healthcare worker, which is associated with that there might be a bare minimum facility, like just a single room and some supplies. And then comes tier 2, which is a district hospital. This is when you have technicians, you have doctors, and you have primary care physicians. Tier 3 is really provincials, where you can really do specialists and you can have good care. And then, of course, national referral hospitals are 4 or 5 hospitals in the country that only tackle really specialized cases. Anytime you're thinking about a disease, and I think what's fascinating is below the 0, I would argue that after COVID, there is a layer -1, which is home testing. And home testing has always been around, but the markets have never prioritized it other than pregnancy tests and some of these tests that require some privacy. But now there is a significant amount of effort in thinking about home testing. And home testing is something where there is no healthcare worker. You yourself are determining whether you want to do a test or not. And the first thing to remember in this context is just making sure that you know which tier you are designing this for, because a test that you would design for a provincial or a national lab will not be the same as you would design for a primary care or a community health worker, for example. There are a couple of organizations that have sprung up that have been remarkable in terms of helping people navigate the complexity of designing a diagnostic test and also helping you through in the kind of validation studies that are needed. One of the ones that I like quite a lot, and there is a lot of literature on, is FIND. FIND is really an NGO that does not create its own diagnostic tests, but it only helps other entities bring diagnostic tests to market. So for example, for malaria and schistosomiasis, last year they ran a competitive program where they took other people's tests and then they helped do independent evaluation of these tests. And what that allows people to do is, if you don't have expertise in a given area, they will choose doctors in Indonesia, doctors in India, doctors in Ghana, wherever you have a market, and they will let them run independent validation programs. And then because they have done a lot of work with WHO, they help put together a package that could be put in front for, WHO doesn't provide a stamp of validation, but WHO does provide a stamp of approval. And so they're a remarkable organization. There are a couple of organizations like this as well, but FIND is really fantastic in terms of just the breadth of work that they do. And of course, COVID was very similar, but they do this for a very large number of other diseases itself. I think, of course, because diagnostics became such an important issue, there is a lot of work that happened in this space for COVID itself. I don't know, I'm assuming all of you have seen what EUAs are, the emergency authorizations, which are these temporary authorizations that were given to these RDTs or many of these diagnostic tests, which will now be going back for the final approvals itself. But it's valuable if anybody is picking up a diagnostic project to actually take a look at the FIND website. They have calls for different sets of, essentially you can think about different diagnostics, and they will call for entities that are at different scales. Either you are just starting to design your test, or you are really far ahead, or you are really in a clinical phase. And they'll help you validate your test in the field, for example. So on the design side, I think one thing that we'll dive in is just very briefly a little bit of a review of what types of things are at play right now. Of course, one thing that's really fun about this space is how interdisciplinary this is, because you could be thinking about there is handling of samples, there is the class of the molecular biology that you actually need. You need to manufacture your reagents. There is a lot of fluidics involved in this, handling of these sets of samples. Some people have already discussed flexible electronics, which is a field that's exploding currently, printed batteries, printed communication. There is a lot of interest currently in self-reporting, where the test actually do tell a positive or a negative rather than folks essentially reading, so no human readouts. And so I think on the technology space, there is a very large class of things to play with. And one of the things to think a little bit about would be is comparing the approaches in which you are building and designing centralized diagnostic tests versus distributed diagnostic tests. I'm a little bit biased towards these distributed tests, primarily because the goal is for these to leave different lab conditions. But depending on what you're interested in, you should really do a Fred Park on that itself to evaluate whether you could do something in a centralized manner. So TB is a great example for this. For TB, in India, there is a centralized context where anybody that is found suspect of TB, the samples are sent to a centralized facility. And you can see that there are certain sets of delays, but on the other hand, the quality of the test is standardized. So you can often think about this, that many of the discussions that we're having so far are really around distributed tests, but there is a framework where centralized tests might actually be much better sometimes. And this is given by the type of a problem that you might be interested in, for example. In terms of technologies, I think we will play with this a little bit in the class. And I think one thing that might be useful, Tyler, is depending on either you or for some of the products, it would be fun to bring some printed electronics to the class. There are lots of printed antennas. I'm sure all of you have seen RFID tags. There is a massive space in terms about thinking about printing functionality on paper that can couple with diagnostics. So up till now, most of the things I talked about were biochemically detected, but there is a lot of interest in electrochemically detecting and reading out. And again, I think much of this is around trying to reduce the human error. And I'll give you a really fun example of the work that we did in COVID, where we found a very fundamental human error that was prevalent in a type of an assay to an extent that we had to completely change our assay because, we knew this, but we learned it the hard way that, 1 in 10 males are actually colorblind. Such a simple thing that if we had just believed in the statistics, we might have redesigned the test from early on. And we learned it the hard way that many people that when I look at those colors, and I'll show you the actual data in a second, to me, it's like, it's night and day. I can tell this color apart. And when we did the field test, we learned that many users had phenomenal difficulties in being able to tell those apart. And again, I think the biggest lesson to learn there is if you can automate certain aspects of readout and make them quantitative, you can remove a little bit of this human context around readout. And although the amplification technologies have evolved significantly, the readout technologies still are not at par. Currently, the primary readout technologies that are at the best level are fluorescent based, and then they require too much infrastructure. So I think one big space to think about if anybody's excited about a diagnostics project, which I would be excited about exploring the space of electrochemical detection of nucleic acids, for example, where you are not relying on a pH change or magnesium concentration change or a colorimetric readout, but a direct readout of amplified amount of DNA. And there's a couple of ideas that Anton and I in the lab have been tossing around using surface chemistry in paper to detect bound DNA. So I'm assuming most of you have picked up projects, but if anybody is really interested in thinking a little bit about paper-based diagnostics, this would be a really fun thread to build upon. Lots of people in COVID and prior to COVID have done cell phone-based readouts itself. Again, they have their difficulties. One of the challenges associated with cell phone-based readouts is the lighting conditions. Many companies did come up with apps, but you can see that you can fool the app by just you take a picture in a different light. And it's fascinating if you look at the BNX Now assay, which I'm assuming, has everybody done the Abbott COVID test? Can you guys mention a thumbs up or a thumbs down if people have actually done the BNX Now test? One thing that is fascinating is go back and open their what is called IFU, which is instructions for use. And I'll talk about the importance of IFUs in a second. Take a look at what they call a negative and a positive test, which is what they say is the faintest of line is still positive. And you can even the picture that they present in that test, you can look at it and you'll be like, I can fool myself into seeing that readout. And I'm talking about, this is the biggest company, the most amount of funds that were pumped in. And of course, they made a lot of money on those tests, but it's still not foolproof. The fact that the most common RDT test is not foolproof. Forget about the sensitivity specificity, which has its fundamental limitations. Even the sets of tests that were rapidly rolled out at large scale readout still remains a big challenge. And I think this is why I'm kind of excited about this space of technology. There is a lot of work that has happened on printed electronics, printed batteries, printed displays, thinking a little bit about degradable electronics, because clearly it's not neither environmentally friendly nor cost effective to do disposable electronics, which is the direction the industry went in. So you can look at many $50, $35, $40 tests where you use them once and you dispose the electronics, but all of that electronics is essentially electronic waste. So if you were to think about, hey, we're going to implement a program with 300 million of those tests done, that is just an absolutely non-starter to begin with, both in cost and the environmental impact. And so very much in the conversations that we had in biomaterials kind of a story, diagnostics falls into a place where if you're going to build use and throw tests, you really have to also be thinking about the environmental context. And unfortunately, because samples can contaminate other samples, you cannot really reuse substrates. Reuse is a little bit tricky because, we are talking about contamination. Although, for the last 100 years for malaria microscopy, we do reuse the slides. The same glass slide that you do a smear on and you do the image, the second day you just wash off your smear. So malaria tests currently using microscopy are 100% renewable, other than the chemical reagents, because even the glass slide is reused. So I think some of you who are interested in the cutting edge of this, there's a lot of space in terms of thinking about what does the future of these sets of diagnostics looks like. I'm going to switch gears and just tell you maybe 5 minutes on what we did for COVID, which was basically my life for 2 years on the bench. But before I do that, I want to take a few sets of questions. Any questions? Because this might look like a very dull lecture otherwise. So I'm going to pause. If people can turn their cameras on, I'm just curious a little bit about what comes to your mind in the conversations that we've been having. I see certain sets of chats. If people could turn their cameras on, and just any questions you might have about what we discussed before I dive in to tell you just a little bit about challenges, firsthand challenges of developing diagnostic tests at scale. But before we do that, I'm seeing at least some of you are in class. Any questions so far?
Can you talk about the biggest challenges to making these at-home diagnostics in materials that are not plastic?
Yeah, that's a fantastic question. And I think we last year or last to last year, when we picked that up as a project, I couldn't find anything fundamental. So it's actually quite strange that the industry has been using that, because we think environment cost is free, ironically. Yes, you have to have some waterproof, but in the lab, we looked for several materials that have waterproof coatings. I think one of the things to think a little bit about is humidity and how these tests do in humidity. But on the other hand, you already do have a paper strip in them. So it's not as if they have certain sensitivity to humidity to begin with. And actually, we couldn't find anything fundamental other than, as a designer, you have to put that as a first principle. And if you do look at the Abbott test, the BNX now, it is actually a paper card. They do try to minimize the amount of plastic in that. And that's the first test I have seen explicitly that did try to actually minimize. But then again, sometimes you have to think about a Titanic. Like the robots that I showed you are the baby versions of actual assembly machines. So if you happen to have a machine that already uses that same module and all you're doing is changing your reagents, it's just much more easy to scale up the factory and capacity. So a manufacturer does not think as if that if you're going to start a new developing a new test should not mean a new manufacturing plant. And so now when we think about, there's a lot of conversation currently around building in South Asia, Latin America, and Africa diagnostics capacity by implementing on the ground manufacturing. And this would be the time to not just implement the same sets of machines and capabilities. This would be the time to essentially revitalize the types of instruments that are used in the assembly lines. But I don't think there is anything fundamental other than that we should assign a cost, an environmental cost, to single-use disposable medical supplies. And I would say almost 50 years ago, there was a starting transition in general in hospital supplies that everything started becoming disposable to reduce the amount of infections in hospitals. And that took that turn that now literally almost all sets of supplies, if the doctors can, they do want to use them as a one-time, because the environmental cost is almost zero, while the cost on the patient is high because infection, a hospital-caught infection, can be very dangerous. So, I think it's a challenge. And again, going back to, remember in last class, we discussed the idea around ethics of design. And this is an ethical challenge, that how do you balance the fact that the test quality might get reduced if you do. And I think one big thing that we've been discussing, which was also a project last year, is having a library of biomaterials that is accessible to designers. So they don't have to come up with a new material for a new solution, that they could just borrow from libraries that already exist. Any other question before we dive into kind of COVID from the front lines?
Maybe
Yes.
I'm wondering about how much instructions vary from test to test, So they seem to have the same principle, but here in Germany, they give us like 3 tests per week if we want to, of course, but they change provider every time. So if you pay attention to the instructions, they vary in the amount of seconds you need to put them in, or the amount of drops you should use, or I don't know if you should cut it or not. So I'm curious about how this really affects, because people usually don't read the instructions several times. They just read it once and follow it.
Yeah, I mean, that is a $1 million question. And Diana, I'll tell you in the next topic that I'm about to cover on a COVID test, designing the IFU and the instructions for use for an average person was the hardest part of us designing the test. And we designed it multiple times. And I don't know if I have the multiple versions, very subtle changes. And we did usability studies by just replacing one word with another led to completely different outcomes. And so this type of thing can only be done by pilot testing. So when you read an IFU of a test, FDA, when you submit a test, you also have to submit an IFU and you have to submit usability data. And that is an incredibly important, and so any of you who's building and designing frugal science projects, you really have to think about usability. Any instrument that you build, if people can misunderstand your instructions, they will. And just for kicks, if anybody has not seen the YouTube video of how to make a peanut butter and jelly sandwich in programming language, do any of you know what I'm talking about? Okay, this is, okay, at least Tyler knows. Tyler, can you find that video and post it on chat? Watch it at a later time. This is the power of instructions, that when you read, people will assume what you said is what they do, and then it's lost in translation. And this is also one of the key reasons why implementation of diagnostics is hard, because many a time the designers that are building these tests do not have any context, any language perspective, the words that you would actually use when it's translated to a different community. So you can't just take a COVID test that's all IFUs in English and ship it to another place and assume that it would just work as well. And I'll talk a little bit about what goes in designing a good IFU in a second, and how to design a usability study. I mean, I'll give you one example of what we did. Any other question before we jump into sort of our own work in COVID testing? How much time do we have? Do we still have 10 minutes or so, Tyler? Okay.
Yeah, we should. I think we currently go until 45.
Okay, yeah. Any other question before I dive into COVID? Okay, I'm not seeing any. So I'll tell you a story that unfolded in a year and a half. I'll keep it very short because I just want to show you the gist of it and lessons rather than the technical details. So this is a test that I've been working on for the last year and a half, almost 2 years actually now, since the start of COVID. We call it SnapDX. The idea was we had been working on a malaria test for the last 5-6 years before COVID around isothermal amplification assay. And many of you have heard about isothermal amplification. If you all do a color test, I'm assuming everybody at Stanford is regularly doing a color test. It's basically an isothermal amplification assay, LAMP assay. And we had been building and designing a malaria multiplexed assay, which would not require, or actually, which would be viable in the field. And then when COVID hit, I was seeing a lot of work in PCR, and I think this was not just, many people around the world realized that we need to use something simpler than PCR to translate it outside the lab setting. And many groups around the world started designing isothermal assays. As a lab, one of the challenges that we put on ourselves is, can we build and design a home test that you could do that is electricity free? That was kind of our challenge because we work with community health workers, and we wanted to be able to literally untether this type of a test, very much like a lateral flow assay test does not require electricity. Could you do a molecular assay that doesn't require electricity? So if any of you want to read a little bit about this, you can go to this website called snapdx.org. The clinical validation study is still ongoing. If any of you are interested, you can click on a consent form. You can actually provide samples. Some of you who are in the call, I've actually reached out in the past for samples, and thank you for engaging in that. So first of all, it was a large team, and Anesta in my lab and undergraduate Abby in the lab engaged really early on. 2 postdocs, Shalab and Adam in the lab, were engaged on assay design and kind of working a little bit on handling saliva. And then a couple of clinical folks engaged. One of the factors was really the clinical staff. And one of the big things that we focused on is, could we take a test, build around 5,000 of those tests, and really run usability studies to really understand what does it take to scale a test. This is sort of a quiz for you guys, but it would be useful actually. Let me just ask. So you have all heard about COVID tests. Could you think about, you can type your answers in the chat. Roughly for the last 2.5 years, we have done 3 billion or so COVID tests in the world. And then the question to all of you is, how many or what percentage of those tests were done in low and middle income countries for COVID? So what percentage of those diagnostic tests? You can type your answers in the chat or just yell. You can unmute yourself and yell. So, I'm not saying low income countries, I'm saying low and middle income countries. That's majority of the world is low and middle income countries. So anybody wants to put in some numbers? I'm actually quite curious. And I said 3 billion tests have been done. So 25%, 20%, 20%. yeah, kind of people are around, floating around 10 to 20%. And shockingly, the answer is 0.4%. So, when you think about, oh, we have a new variant coming out, this is a no-brainer. Why do we have new variants coming out? Because we're not diagnosing at all. And one of the threads to think about is when you really let something like this run rampant. So, this is kind of the harsh reality of 3.2 billion tests done total. This is a paper from 2022, early 2022. And 0.4% is the total number of tests. And I think this is kind of, in some sense, in thinking a little bit about why frugality actually matters. So we have some clinical partners for this work in Ecuador. The average household income in Ecuador annually is around $400. And to get one PCR test in Ecuador, you have to pay around $100. So think about, take your average annual income and think about 1/4 of that going in a single test. And so no wonder we haven't been able to scale anything. And this was kind of at the heart of thinking about, and very much like I told you all the untold story of Foldscope, the SnapDX, the design criteria was we want to do a molecular test, but it has to cost less than $1. That was our price point. Like, can we do it? Is that feasible? While currently in the market, there are these assays, molecular assays that can cost anywhere from $50 to $75, for example. So we wanted to reduce that by 2 orders of magnitude. This is the setting that we are interested in. As I had mentioned, ASHA workers, this is literally when the Delta wave was happening. So these are ASHA workers running home-to-home COVID screenings in India. And you can think this is the kind of a setting that we are interested in. So these are highly trained professionals. They don't have big degrees, but they do this job day in and day out. So they're trained in the sense that they do this repeatedly. This is kind of, I'm not going to go into technical details here, but one of the things that we ended up inventing is a new way of thinking about how to build a little syringe. And if some of you will the lab session, we will bring a whole bunch of these devices to show you, in which there is a glass capillary inside. And that glass capillary is what our cartridge is. And this is literally everything that goes on. And if some of you are interested, we will actually make you assemble one of our units. We built 10,000 of these, at least supplies for 10,000. We finished assembling around 5,000. And it was a massive ordeal in terms of thinking a little bit about how to do this without electricity. We primarily use hot water. There's a lot of material testing going back to materials. We could not find biodegradable materials that would be compatible for these. So most of this is actually built out of Teflon and high-grade plastics. But that's the unfortunate current reality because the temperatures are still 60 degrees Celsius. We put together an assembly plant, and some of you might have come by and seen this. This was built in Sri Ram in the teaching space, where we assembled around 6,000 of these units. And then I want to go back and respond to kind of Diana's question. This was a fascinating story. So this is the intermediate IFU that we designed. Anesta in the lab worked quite a lot on this because we early on realized when we started giving the IFUs to people and started shipping the test in the Bay Area, and we would record some of our members and we would see that they're doing the completely wrong thing than what we want them to do. But again, remember, these are people that have never done any science. This is an average person running a molecular assay. So we did 3 rounds of this usability study. And here are the results, which is actually quite powerful in terms of thinking about why usability study matters. In our first study, we had maybe around 65% of people were able to use it as we had designed. Then that number went to close to 70%. Then it crossed around 80%. And in the last session, it went all the way down to 95%. And all that we did between these usability studies, this is roughly around 400, 200, 500 people, was really just work on the IFU. Of course, we redesigned certain aspects of the device itself, but IFU was completely redesigned. And this goes back, and again, whenever we had conversations with the FDA, this is actually a very important criteria. The other data that I want to show you is on the quality control side. There's a lot of machines that we ended up building that primarily were quality control machines. One of the big things about diagnostics, and again, this is why we wanted to make roughly around 5,000 to 10,000 tests, is if you're just writing a publication and you make 10 tests and you publish that data, that's okay. You don't actually understand the variability that might be in your process. So this was a fascinating exercise to make sure that we can do some amount of QC. So this is one of the instruments. There are a couple other instruments that we built around quality control. And then the one thing I want to show you around color blindness, this is something that we learned along the way. This is another color blindness or color identification study that we ran. We were using a colorimetric assay before, in which you were supposed to tell whether it's pink or whether it's yellow. And now if I give you the spectrum all the way from one end to the other, most of you will be able to tell, but there might actually be someone in this room literally right now that is not able to assess this correctly. And the scale that we previously had used fell into that radar that a significant portion of people in our study mislabeled the samples, to an extent that we decided to actually use a different readout mechanism associated with this. And that was a big lesson in the end that we could have learned if we had done this usability study prior to running the entire thing. So we had to redo the entire study primarily because we had changed something fundamental, which is, again, with diagnostic tests, it's a pain. If you change one thing, all data you have to retake again. So, currently we are planning to ship around 1,000 of these units to Guatemala. We have a collaboration with the government and a few NGOs in Guatemala to train community health workers and do the same thing that we did for Bay Area users, for example. So this is an unknown Bay Area user using the test, someone that we've never met, but do the same thing, but with community health workers in a field setting. So this is kind of what we are gearing towards. And one of the things I want to end this on is, first of all, of course, it was a really wild ride going from the kitchen table, this is my kitchen table, to getting a test in a hand of somebody in 90 days. Although we could get that, it still takes almost a year and a half or two to really build a product, which we kind of feel where every aspect of that product was designed from scratch. To an extent, now we can manufacture these cassettes for roughly around 40 cents. We can manufacture with open source reagents. We can literally make reagents in-house for roughly around 30 to 60 cents. And so we've met that bill of materials type of a criteria of around $1 in manufacturing costs. One of the fun things of the reason I'm really excited about this is the framework that we built allows us to do saliva, sputum, urine, and blood. And these are the samples that we are after. Saliva for respiratory viruses, sputum for TB, urine for schistosomiasis, and blood for malaria. And one of the interesting aspects is that using that exact same platform, we can essentially swap cartridges that I was talking about to switch. And this is also the power of molecular tests that you cannot do with antigen tests, that literally just changing the primers. So if a new pathogen came along or a new strain came along, you can actually detect it by just changing your primers. So this is where we are. I'm just putting together this paper finally. I think once I finish it, probably sometime in mid-May, I'll share it with folks if people are interested in reading much of the details associated with this. But kind of one thing that I want to comment on is this has been an incredible labor of love in some sense of really thinking about what does it take to design a test from scratch. And then again, keeping this notion in mind of doing these things at this intermediate scale. You know, if we had done just hundreds of units, we would not have discovered the kinds of fundamental problems that we had to solve. And there are still several other problems that we still have to tackle, include lyophilization and kind of shelf life at 40C, for example, which is what we are targeting for, because for malaria and TB, we need to really make that shelf life to be stable as well. But I think, yeah, that's all I want to say about SnapDX. There's a lot of details in there, but I'm realizing that I'm already running out of time. If there are any questions around SnapDX, we can take that. But Friday, we will do a deep dive on it, because I'll ask Anesta to bring a few units. If some of you are interested, I mean, one of the really fun things we did at Stanford is I had a call and we got somewhere around 10 to 15 volunteers, and we trained them to assemble the units from scratch. And so we had a little bit of this small scale factory. Now we're trying to roboticize it, but it was kind of interesting. And so if you're interested, you can actually on Friday build a test from scratch all the way. And it'll give you a sense of just complexity of what goes on in terms of the number of parts involved. But I'll stop here. Any questions / comments, and then we will pick up that thread also in a lab session.
So it is still not possible. Oh, sorry, Tyler. It
Go
is
ahead,
still
Dan.
not possible to get one outside of the US for
So
the SnapDX.
this is not a product yet. This is still in research study. We're just publishing our first validation study. And then we are working, I am planning to launch this as an entity. So that would require then building an entity and scaling up. I mean, there's many, many steps involved. We're really in the early phase, but one of the power of this tools is, of course, we built this in the context of COVID, but the demand for molecular tests for a very broad group of pathogens is really why we built it. So I think, yeah, you know, I would say maybe we are a year or so away. I think around a couple of months from now, the plan is to launch this as an entity, but then it still takes a year or so to build the kind of manufacturing capacity and redo the amount of validations, but for a broader group of people. But our next scientific question we're asking is, can we demonstrate that a community health worker anywhere around the world can run the test without electricity, just using boiling water? And, you know, we demonstrated this, that can be done for Bay Area users, randomly chosen. We have demographics of all those users, so it's very broad, but it's still not the same. And our focus area, one of the focus area is definitely community health workers.
So in principle, with this last comment, so you will be focusing more on this SnapDX being used not at home by me, but myself. But
No, I think it's all modes of applications will be open. Scientifically, we are trying to demonstrate that community health workers that sometimes might not have a high school degree are perfectly capable of running a molecular test. You know, that is a question that many regulatory bodies are asking us that we need to answer scientifically. I have a very strong belief that that might be true, but we have to prove that scientifically. So that's the purpose of that study. But again, in many markets, at-home tests are fundamental. And I think, you know, for many sets of diseases, privacy is so important that it might be that an at-home version of that is useful and valuable. But on the other hand, at other places, community health workers are really the ones that are providing a service, and just knowing what you have is not of value until you can also get a cure for it. So there is a balance between those two approaches. And I think just like Foldscope, one idea is to launch that in the dual markets. So use the at-home markets in US, Europe as well to actually support financially what we do in other markets. Anyway, these are all plans that I'm structuring right now. But I think our Guatemala connection and a connection in Bolivia has been very interesting because they've been able to push us on the kinds of conditions, especially test conditions like weather-wise. And that's been one of the hardest part of this project, is making sure that we can really build something that's room temperature stable and will survive the kind of abuse in transport and other things. Any other questions? Yeah, and you'll all get a chance to see it on Friday. So I think, yeah, just it'll give you a better sense physically. Yeah, because I kind of skipped over how it works just because of shortage of time. And I think going back to, I think Emily had asked it, Anton and I had many discussions around this. It still is an open question. For slightly higher temperature, many molecular assays go at the least to 60 or 70 C. Eppendorf tubes around the world are a massive disposable plastic waste that scientists make, but there are very few plastics or very few degrade. Actually, I am not aware of any that. Reuse is one thing you can do in research, but in diagnostics, that's very hard. Currently, we collect the device back because we want to do other things with it. But yeah, I think that's kind of one of the next tough challenges to think about for molecular reagents. Could you think of materials that are capable of handling those temperatures and are still degradable? And yeah, I think we've talked about it, like, there are many materials in nature that would easily do it. We just don't know how to make them. Like abalone's shell, for example. And also it has to be compatible to biological reactions. Okay, I think it's already it's 2 a.m. here and I need to sleep. So I'm gonna say goodbye. I'll see you guys in 2 days. Sorry for the connection, and then we'll catch up. I think one thing to remember is next Wednesday, we will have our first Teach Me Anything session where Susanna from the class will talk about how to make vaccines and why is it so hard to make vaccines. If many of you remember, she's a faculty in, I think NYU in New York, I'm trying to remember, but one of the medical schools, and she's dedicated her career on making HIV vaccines, and she'll talk a little bit about why it was so hard. And then depending on if permitting time, I'll do a little bit of statistics for all of you to get an intuition for the picture of vaccine inequality. Just where do we stand today? What needs to happen? We think the pandemic is over because we have access to vaccine. That's not the case for majority of the world still. And what needs to change to something like that to happen? So we'll discuss a little bit about vaccines. And then the lab class will really be around diagnostics. One thing, Tyler, if you could write on Discourse, is everybody should go find used diagnostics kits. And if they are not so expensive, you can bring the fresh ones so that they're not contaminated with people's samples. If you do it at your home, you can do a breakout session with whatever contaminants you have with your nasal swab. But if you do it in the lab, bring a fresh one so that we don't have a super spreader event. I think actually, no, maybe they're extraction buffers. It's possible that it's inactivated. We do a lot of work to make sure that the saliva samples are inactivated by heat. I don't know whether RDTs currently have a chemical buffer that completely inactivates. Somebody should check that, actually. That would be useful to know.
I'll take a look and post on the Discord, yeah.
Yeah, but I think, yeah, people should at least bring a whole bunch of RDTs, and I have some other electronic tests that I'll give an ESTA. Okay, on that note, I'll say bye, everyone. I'll catch up next week. And again, sorry, I'm away for a little bit, but we'll do some of these online, and I'm hoping my connection will be better next time.
Thanks, everyone.